few months ago there was a controversy over the testing, and specifically there were a bunch of people who felt that since they didn't have a known mutation, they felt they were being excluded from being studied and therefore less valued. And so we ended up getting into a very long discussion about what the purpose is of the various studies, and what kinds of information you get from various studies, what kinds of things can go wrong, and so forth. And we decided to turn that into this session. And so, so I'm going to be doing this together with Dr. Lehmann. And the role of Dr. Lehmann in this session is clear because he's the one who does the studies, so he's the obvious expert on that. My involvement comes in from various aspects, which you'll see as we go along. But one is that I'm one of the doctors who's most active on the listserv, and therefore I know some of the history of what's on people's mind. I'm also involved in some of the process of using the information, in that I run the largest database that's used for neurologists to help make diagnoses. I'm also involved with Dr. Lehmann in working on finding one of the new genes that accounts for some of the people with hypokalemic periodic paralysis who don't have a gene that's currently known. And also I served for many years on the institutional review board at Harvard that determined what studies got approval to be done and so forth. So I'm familiar with it from that end. So let me start out by framing the issue that came up. So the way we're going to do it is I'm going to talk first about the kinds of issues that came up and people were concerned about. I'm then going to explain some of the various types of studies. Then I'm going to explain a few of the things that nevertheless go wrong despite us having tried to organize science and medicine in ways that seem reasonable, just so you'll understand it's not just you, that some things go wrong despite good people trying to do good things. And then Dr. Lehmann will explain a little bit about how studies are done. But the core part of the session is meant to be interactive, so you're really encouraged at any point to pop in questions and start a discussion. Most of what we prepare is really meant to be a framework for discussion, so don't feel you're interrupting. The whole purpose is to interact. So we'll start out on the next slide. And so the two basic types of studies are, one is you take people who are known to have a particular mutation, and you do studies involving them. And the second type of studies is you take people who are felt clinically to have a particular disease, and you do studies on them, and chief among those is trying to find what the gene is or other cause if it's not a genetic cause. And so obviously if you have a gene already identified, you're in one group. If you don't, you're in the other group. And there's very important things being done with each group. So with the ones for whom a gene is already found, there's a whole bunch of things that can be done for various reasons. One is to characterize what are the various things experienced by these people. Do they have attacks provoked by cold? Does tickling bring on attacks? Or all sorts of things like that. Well, that can be very important in terms of figuring out, helping doctors make a diagnosis by having all the information available. And some of that information gets put into articles. So for instance, the patient groups were very important in getting certain types of information out that weren't really well known, such as with hypokalemic periodic paralysis, that a lot of people have their attacks triggered by sodium chloride, which most neurologists have no clue about at all. It's not included in many of the review articles. but it seems to be a very common thing. So that type of information, getting that documented, getting that out to the doctors so they can make the correct diagnosis is very important. That process occurs in two ways. The sort of old-fashioned way is teaching people, putting it into review articles, textbook chapters, and so forth, and getting it incorporated into sort of lists that people look up are important. The direction the field is moving in now is actually having various type of diagnostic databases. And my day job at the moment is at the company that I started called Simulconsult, where we have several thousand diseases in a database, and we try to help doctors put together that information in some very sophisticated ways to try to make the diagnosis. And so at some of these, at the last meeting 2 years ago, we added in a whole bunch of things we didn't have before. And the nice thing about databases like that is that the delay between a discovery and it being used by doctors is basically zero. And you don't have to have, for something like that, you don't necessarily have to have a big formal study. You could have, like there was one thing, we just had people raise hands in the room, and we figured, okay, it's about half, and we could put that in as an initial approximation. Obviously, it's best to have a published peer-reviewed study, but in the absence of it, one can proceed with information. So what these type of studies are doing is to try to systematize that information. And it's important not only for diagnosis, it's important for treatment, because if you can figure out that somebody has a particular variant and do a gene test and so forth, then you can find, okay, these people should be treated with this medication or that, and should be careful about this, and it becomes very important, and sharing this information becomes very important, because I'm sure a lot of you went through a long process of people trying to figure out what you had that may have lasted years. So these are often referred to as diagnostic odysseys. And so one of the goals of this type of research is to shorten the diagnostic odyssey. The second part of the goal is to improve treatment such that instead of people trying things that aren't optimal, the more we can divide people into subgroups, the better. And some of that is going to be on the basis of the exact type of gene mutation that you have, but others are going to be other types of things. And one of the issues that has been discussed back and forth on the listserv, and maybe Dr. Lehmann will be able to give us some extra insight beyond what we've been able to achieve on the listserv, is the whole question of digestive disorders. So people with hypokalemic, people who are known to have a diagnosis of hypokalemic periodic paralysis are more likely to report digestive problems than other people. And as far as I can tell, there are two possible explanations for this, and I'm not sure which one is correct. One is that there's just something about the gene that causes a problem with digestion. The other is that problems with digestion tend to cause you to lose potassium into your gut. And therefore, somebody who has probably 10% of the population has some sort of problem with their digestion. So the 10% of the population, so you would imagine just by chance that the 10% of people with the hypokalemic periodic paralysis gene would have a digestive problem, and those are ones who are more likely to come to attention. So those are some of the types of issues that we're trying to get to the bottom of in a lot of these studies, and that's why the studies of people with no mutations are important. But I think the discussion on the listserv was from the other perspective, was people who don't have a known mutation saying, why are you ignoring us? And we aren't, basically. In fact, my involvement has been entirely from the point of view of the 30% of people with hypokalemic periodic paralysis with no known gene, because we think we're going to be able to pull another gene out of that group of 30%. And furthermore, we think that that's going to be related at a molecular level to to attention deficit disorder, so we think that it's a sort of a tiny tip of a humongous iceberg. And so nobody should think that they're being ignored because they're not known to have a gene. And one of the things that we're trying to do with some of the things like testing for lidocaine sensitivity is we're trying to take the 30% with no known gene and say, what can we do to try to get homogeneous groups that would allow us to say, the more you can break people up into groups that are different from, in terms of what's caused the problem, the more that you have a homogeneous group that you can work with and do genetic studies and find genes, and even before you find the genes, find different approaches to prevention and to treatment. So nobody should feel ignored if they're in either of these groups. These are the kinds of things we're trying to do in both of these situations. And one of the other things, I guess many people who've been on the listserv are going to be aware of this, is we're not just trying to educate, we're not just trying to collect this information to educate doctors, as many of you have found. A lot of doctors are unaware of this stuff, and it's difficult to get information to busy people. And one of the things that we're trying to do is to empower you as people who have the problem to be the ones who will help educate healthcare providers. And one of the things that came out of the listserv was people said, we have all these questions. A, we want them answered, and we want them answered in an authoritative way that we can take to our doctors to help bring them the knowledge. And the issue is that doctors are sort of sometimes overwhelmed by patients coming in with a huge pile of information to say, you should read this. And somebody even told a story that they, so what we did was we wrote up this, so people on the listserv came up with a bunch of questions, and Dr. Lehmann, Dr. Yurkat Rod, and Dr. Levitt and I wrote up something which we called an owner's manual for hypokalemic periodic paralysis, and that's up on the web, and we can help you find that. But somebody told me that they took that into a doctor in an emergency room. The doctor just tore it up in front of them, which is sort of a little shocking. But we're working on ways of dealing with stuff like that also. And the Child Neurology Foundation is actually thinking of taking this innovation that we developed on the listserv to make a series of owner's manuals for diseases that basically the doctors need some help on. And the idea is if we do this sort of branded approach of people will know, oh yeah, the ones on the Child Neurology Foundation website are good ones. And so the idea is to try to take this innovation that came out of the listserv and turn it into a much more widespread phenomenon. And I'd like to add that the listserv has been terrifically innovative, and there's been a bunch of terrific people on it who've really, it's like having research assistants, and it's like being able to poll people and get answers on things, and it's an amazing resource. And there may be other things like that in other areas of neurology, but of the things that I've seen, this is really the most vibrant and well-functioning resource. And it's a tribute to the Periodic Paralysis Association that it works so well, and it's a model for many other things. And people are doing a lot of things in terms of healthcare reform, but it's sometimes the things that you're not thinking of that are tremendously effective of this social networking type, like the listserv, that's important. And this has driven off a lot of the information that we both are sort of doing these quickie studies on the listserv, organizing people, feeding people into studies, bringing back information from the study. It's a crucial part to all these studies. And so it's one of the ways that things really work well. So I wanted to emphasize that point before having the next slide of discussing things that don't work well. So let me just point out a few things, just because people are inevitably going to be frustrated about things that don't work well. So one of the chief pillars of medicine as it's organized now is to treat people according to evidence-based medicine based on randomized clinical trials. And it's a terrific impulse. It's absolutely the right thing to do, but it sometimes leads to results that are basically wrong, or I think that the outcomes are strange. And I want to explain how a few of these things are dysfunctional, because you're going to run across these, and I want you to understand where they come from, because when you find something that seems weird, you should at least understand why it's happening, and that the more we understand it, the more we can develop workarounds, and the more you guys can feed information to the doctors, and the more the doctors can help empower you to get around these. So I thought I'd give a few examples, and doctors don't usually do this because we don't like to sort of give examples of how things sometimes don't work, but I think it's really an important part of empowering people that you should understand at least as much as doctors do about this, and probably more, because doctors typically just think of their role in the system and they don't think of the system as a whole. One of the things that goes wrong is that a drug will only get studied in detail, in enough detail to get a big randomized clinical trial if it can be patented. So that leads to all sorts of weird things. So there's a bunch of instances in which the group of drugs called methylxanthines, which are present in coffee, tea, so it's like caffeine-like drugs that are in coffee and tea and chocolate, are very helpful in a variety of conditions, including asthma and attention deficit disorder and so forth. And they are not, they are typically not recommended because there haven't been randomized clinical trials, although doctors are aware that they actually seem to be as efficacious or better than many of the drugs that are tried. And so people just sort of recommend it, and one of the examples that I'm pretty familiar with is attention deficit disorder, that typically it's said to sort of go away when people get to be adults, but largely what happens is people start drinking coffee, and that ends up being a treatment that's as good as Ritalin or other treatments that are being used for attention deficit disorder, and it's sort of easier to get with a lot less, and has less side effects and so forth. So that's one of the sorts of things that you have to be aware of. That those type of things will often occur with more common diseases. With less common diseases, as hypokalemic periodic paralysis is believed to be, there are other types of problems that can arise. And one problem we've seen come up with a whole bunch of drugs in the last decade in neurology relates to the Orphan Drug Act, which is a United States specific thing, but it affects things in the rest of the world just because of the dominance of the United States in terms of drug development. There have been a number of instances of drugs that have been available, and then they get taken off the market, declared as orphan drugs, studied in great detail, and then they come back costing 50 times as much as before. So if this happens with various, a lot of people are upset about this, trying to do something about it, and feel that this isn't what Congress intended. When Congress passed the Orphan Drug Act, the intention was to get things to work properly for drugs in situations where nothing was available. And although it seems to fit within the letter of the law, it's certainly not the intention of the law that the drugs be withdrawn from the market and then put back on, costing 50 times as much as before. But there have been several instances of it already. So, so you should be aware that this is something where people set up these mechanisms, and and these are things that I think the way they set up the Orphan Drug Act was not worded right, but you will see things like this in rare diseases, and you may see things like this happening in periodic paralysis. So you should be aware that this is a potential issue, and this is another way where systems that were set up to be reasonable end up producing some outcomes that a lot of us would feel are not reasonable. A third issue is that it's just difficult. It's difficult to change what people are doing. The way doctors think about diseases, it can take a very long time for medicine to change, even when after there's enough evidence where a reasonable person sitting down looking at the evidence in a lot of detail would conclude that things had changed and one should do things differently. And that even happens in basic science, and I put up the name of the classic book in the history of science that describes how basic science is dysfunctional in that. And it's a fascinating book to read, and it just goes through all these examples of great ideas in science that took a huge amount of time to convince the field, and in some cases it's a matter of just a new generation looking at the new explanation, saying this makes more sense, and just waiting till they get in to be the decision makers. And of course, that one should do better than that. One of the things that we're doing with diagnostic software is to try to make it so we basically, instead of that taking decades in clinical medicine, that could start happening immediately in terms of new information being put into diagnostic software. So we're hoping that clinical medicine will be faster than basic science in terms of scientific revolutions of changing the way people think about things. But largely people do a lot of things in their head, and one of the examples that's most troubling about periodic paralysis is that people have these rules of thumb as to what is what's called at various times a conversion disorder or hysterical paralysis. And so a large fraction of people with hypokalemic periodic paralysis have been dismissed at some point as having a conversion disorder or hysterical paralysis. And I wish we could say that through some educational thing we're just going to make that disappear completely, and we're trying in a whole bunch of ways to do that. But you should understand that it's hard to get people to change their whole worldview as to how things work. And so doctors are people, and part of the solution to that is to have you empowered to say, oh, well, I understand that's a good general rule, but in the case of hypokalemic periodic paralysis, it doesn't really apply, and here's an article from the Child Neurology Foundation that describes that. And so in some ways there's going to be a certain amount of dysfunctional behavior within medicine, and part of the answer is you folks being empowered, and we're glad to help, but it's not going to work without us being involved, but it's also not going to work without you folks being involved. And so part of the solution to these whole set of things about research issues is to have you as partners with us trying to work out all these things that to understand the good parts about the system and how we're trying to get this information and translate it into things that will affect diagnosis and treatment, but also understand some of the things that aren't working due to things that haven't been fixed yet, and with some extra effort with you folks and us, we can get around them, but these are issues. So that's my part of the talk. Dr. Lehmann has some slides that you can go through at whatever speed you want. Do we want to break for some questions now, or do we want to, okay, at least one, and then we'll move on to Dr. Lehmann.
I just wanted, you referred to the listserv several times. Does everybody know who that friend is? I personally don't have contact with doctors who understand this disease. And the listserv has helped me go to my doctors and say, try this treatment. I've had to beg for effervescent potassium. I come from Texas. My doctor says, if I give you that much, that's how much they use on death row to kill people in Huntsville, you know. So they're terrified of doing a lot of things that we find out on the listserv that other members do. Deb is, she's my doctor, in fact. I know it says that we're not supposed to rely on the listserv for medical advice, but all of you people who post on the listserv and give us the advice that helps you, helps all of us. And I don't know how many of you have access to doctors like this. I don't. But I think that we owe Deb and the other people who work on the listserv a round of applause for helping all of us.
I just want to kind of address that.
Sure.
Deb is amazing.
Yes.
She started along with Patrick Cochran. They did the first group, which is her group, and the group split up, so that's why it's a little confusing to everybody. But they're wonderful. She has some of the best information out there. Her listserv is great, and I adore her. So both groups work really well together. In the past, people thought you had to choose, but you don't. So I'm glad you brought it up. And
I think all of this help that we get.
there are
we
two
don't get
listservs.
local.
Deb started one. Hers is hkpp.org. I think she might have changed it. It's still the same. On hers, though, they have changed it to where you have to be diagnosed. And then ours is the PPA.org. But both wonderful lists, and I've been on both of them since '99. But yeah, just want to throw that in there. There you go.
Is Deb here?
Deb is not here. She wanted to come, but she was not feeling that great, and just come out.
More questions, perhaps?
And Don Anderson, too, worked very closely with the group, and he had surgery, so that's why he's not here, because he was registered and planning on coming, but where is Deb in
Calgary.
She's in Canada. Yeah, where is it?
Calgary.
Calgary? And what's her last? She moved, so.
Okay, are there more questions now, or should we, should Dr. Lehmann go through his material and then we'll have more questions?
So the first question is, how can we identify a mutation in a gene which is already known and responsible for a disease like hyperkalemic paralysis? So what we need is a blood sample, and the blood sample contains cells and other proteins and fluid. The cells are the red blood cells, which are not important because they don't have any nuclei anymore. These are dying cells, so after 100 to 120 days they will disappear, they will be taken up by the spleen. So the red blood cells we are not interested in. However, we are interested in the white blood cells because all white blood cells have a nucleus, and all the genetic information is contained by the nucleus. So, and we extract the genomic DNA from the white blood cells. And the next step is, in a certain gene. And a gene consists of introns and exons. And in the order, intron 1, exon 1, intron 2, exon 2, and so on, let's say up to 106 or so, introns and exons. And the exons are important. They contain all the information for the production of the protein. The protein is, for example, an ion channel, but could be another protein too. And the introns, in earlier days, the introns were called junk, but that's not true. We learn more and more that the introns are very important for the expression of a gene, and so this is not junk. But for us, for finding the mutation, we are only interested in the exons, because they translate the genetic information into proteins. And finally, we search for the functional changes of the protein which can explain the disease. And so that means we have to find all the exons, and to find the exons we need so-called primers, primer sequences, which anneal or which suit to the beginning of the exon and the end of the exon. And then by using a specific technique, the polymerase chain reaction, or abbreviated PCR technique, we can amplify these exons. And then the next step is that we sequence these amplified exons. And that means that we base for base, so the genetic information consists of 4 bases, and it's just like a book which is written with a 4-letter code. Here we have 4 bases, and it depends on the order of the bases, and this is the genetic code. So base for base we sequence in this exon, and then we compare if the sequence of the exons is completely normal or if there is a deviation. And then the next step would be to find out, does the alteration changes the amino acid which is included in the protein. So there are many, many changes which are not important because they don't change the amino acid information. So, by the way, we are doing exome sequencing nowadays, and we can find in each person, healthy or deceased, about 80,000 genetic alterations. So everybody has about 80, some have 60,000, others have 100,000 genetic alterations. But only one is decisive for a disease. And that means most of these changes will not alter the amino acid within the protein, because this is a 3-letter code, and the first is very important for the amino acids, the second is less important, and the third one is not important at all. And if there is a change on the base level, it doesn't involve the amino acid which will be produced by the cell. So, next slide, please. Here we can see the polymerase chain reaction, and the first step is when we extract the genomic DNA. this is here, the genomic DNA. This is a double-stranded DNA. And this is not useful for searching for alterations. So the first step is a denaturation. That means to separate the two strands so that we have two single strands, and then we can use the primer sequences for the amplification. So this is shown here. This is a double-strand DNA, and here it's isolated. There are two strands, and then we can use the primer sequences for the annealing. And this is polymerase chain reaction itself. We can amplify these parts. Here is the beginning, here is the end, and we can amplify that by n the power 2 of the power of n. So that means in the first cycle, the first PCR cycle, we can double the amount of this exon we isolated, and with the second cycle, it's an amplification of 4, and for the next cycle, it's 8, and then it's 16, and then 32, 64, 128, and so on. That means with about 6 or 7 cycles of the PCR, we have enough material to do the next steps. That means sequencing, and this is shown here. Next slide. This is the amplification, denaturation, priming with primer sequences. Then I forgot to mention the synthesis of the strand, and then this is the doubling. Here is the second, and then here you have 4 times more material amplified. And for the synthesis of the new strand, you need materials like the primers and the bases and buffers and so on. For the polymerase chain reaction. Next slide. This shows the direct sequencing technique. So the secret is the nucleotide mixture. These are the bases, and around 10% of these bases are replaced by a slightly different base which stops the chain. And the final way is the sequencing. That's the next slide, please. Base for base, and we can here see the 4 bases: C, T, A, and G. And the order is C, T, C, 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 A, C, T, G. There's a G. Okay, and this is the normal axon, the normal part of the axon. The axon is much, much longer, perhaps 200, 300, 400 bases long. And here is the same part of the axon of a patient. And then we can compare the 2 sequences and can see here is a difference. Here there are 2 bases, 2 different bases, so each base has its own color. And here we can see that there are two bases, whereas the normal control has only one base, so that's a normal base. And here is on one allele coming from the father or from the mother. One is the normal, here's a black one, and inherited from the different from the other parent is the base which is shown here in green. So, and this is can be decisive for the disease of the patients. But as I told you, we will find about 80,000 such changes in the whole genome. And the next step is to find out, does this base exchange alters the amino acid which will be produced by this change. And here, this is the triplet which is decisive for the amino acid which will be produced. And the triplet CGC will produce an arginine on the protein level, whereas the CAC will produce a histidine. And here, these are the differences in the protein, that means in the channel. So, and this is a frequent change from arginine to histidine. I told this morning that the S4 sequence contain many positive charges. These are amino acids with positive residues, and arginine is one of them, and the histidine is only weakly positively charged, and this makes a change. So, and this makes a disease. Okay, question?
Yeah, simple question. On the histidine chart, there's the lower peaks, and I believe you said that one of them of the two lower peaks, the black and the green, that one of those was from perhaps the father and one was perhaps from the mother. But I look at the arginine on the top, and they're both, and it's just one and it's tall. Why are those different? Are these not supposed to be the same? And also the allele, is one allele from the mother and one from the father, or is that the wrong terminology?
No, that's okay. It's both. It's a combination of the exon DNA from both father and mother together. And therefore here have the higher amplitude, and here where you have two different bases at the same location, you have just the half amplitude. So this is from father plus mother, and this is from father and the other from mother. Okay? Normally they are completely identical. So, okay, the next slide. Yeah, when can we name such a change a mutation? So the first prerequisite is that the genetic alteration leads to an amino acid substitution, as I just said, for example from arginine to histidine. And the affected amino acid is highly conserved. That means that it's the same amino acid in most species, so not only in manship, but also in the mouse, in rat, in the electrophorus, eel. So that means the genetic information is extremely conserved, among the species on our world. So there are just a few changes. So I think at the amino acid level, between pigs and human beings, there is a homogeneity of about 99%. So, and the question is, when it's almost identical, why looks a pig like a pig and a human being like a human being? And although the genes are almost identical, and for this difference probably is the part which was called the chunk genome responsible. That means it depends on which gene will be read, the code will be read and transferred into the protein. And this depends on the age, and on many things, on the species. So not all genes are used for the production of proteins in the cells. So a heart cell uses completely other information of their genome than a kidney cell. Okay. The next is the highly conservation. And then when can we suggest a mutation, or an alteration, a mutation? All affected family members harbor this genetic alteration, so this is very important. And for that, it's very important to know who is affected and who is not affected. So, and vice versa, all non-affected family members do not harbor the genetic alteration. And another check would be to see the functional changes of the mutations on the protein level. That's the reason we form the channel protein and look for the function of the channel protein. So at which voltage, at which membrane potential the channel will be opened or closed. That means, is there any functional difference caused by this genetic alteration? And the final control is that this genetic alteration has to be absent in a large number of controls. That means when we see a change, it could be a new mutation in a known gene, then we have to check 300 or 400 healthy controls and to exclude this particular genetic alteration in these healthy controls. Okay, I think I should stop here. And questions? To any part, to the first part.
I didn't really want to use that. When you say the affected amino acid and the affected family members, what's your definition of affected there?
The affected.
When you talk about the affected I said that wrong. When you talk about the affected the third line, the affected family members harbor the genetic alteration, and then on the fourth line, non-affected. What's your definition of affected?
Okay, that means clinically affected. So is this a patient or is this person healthy? And then if it's a patient, is what exactly are the clinical features? So that means affected. So probably it should be sick or healthy, diseased or healthy.
So that's not saying that someone who's not clinically affected couldn't carry the gene? Have you addressed that?
These are the problems behind all of that. I didn't touch that because then it's to talk about reduced penetrance and minor clinical expression and all these very complicated things. I think it's not the right place to talk here about that.
Thank you. I don't need, I can show.
In the MDA magazine, they were discussing exon skipping as this new method of going down the sequencing, taking out the one badge, exon 13 or whatever it is, and that there's enough readable material that the person still functions. Is, in the case of the ones you've uncovered, is there enough readable material that if you took out the offending exon, that it could work in periodic paralysis?
Yeah, so the exon skipping is very famous, and for Duchenne muscular dystrophy and also for mucoviscidosis. You have to keep in mind that the proteins which are defective in these two diseases are structural proteins. That means it's a structure like a chain which stabilizes, which has a stabilizing function. And if exons are skipped, then the protein will be shorter, so the chain will be shorter, but will still functioning as something which can stabilize the membrane. So it's not the same, but it has some function. On the other hand, if you skip an exon in a gene which produces voltage-gated ion channel, then the missing part will prevent the protein to get inserted into the membrane, and it will not be functioning. It's not a channel anymore. So that's the difference, and therefore exon skipping for periodic paralysis would be a disaster, because no functional protein would be in the membrane. Okay?
I don't even know. Is it possible for someone to be a carrier, have the gene, not be affected by the disease, or pass it on to their children?
Yeah, that's a similar question as two before. That this is reduced penetrance is quite often in the periodic paralysis, usually in females, in the mothers, that they don't have, can't report about any paralytic attacks, and however, they carry the mutation in this particular gene, and for example, the children can be deceased. Yeah.
When you take down the medical history, does it help you to know where to look on the different chromosomes and the axons and everything?
Chromosomes, different chromosomes.
Does it give you an idea of where to look?
There are some syndromes. There is an aberration of a part of the chromosome, and this is a special technique. This is called cytogenetics. Then you can see a part missing on a chromosome, or from one chromosome to a different chromosome transferred. So this is very helpful. It's a very helpful technique in major syndromes where several genes are deleted. And that means in these conditions you do not have a particular disease, a particular symptom of one issue, of one tissue, for example, muscle weakness. In these conditions you have several symptoms from several tissues. So these are so-called syndromes. And in these syndromes, it's very versatile to do cytogenetics.
Let me add something on this, because this is something that we do in diagnostic software a lot. So there's a lot of times from the clinical situation, you can put together some very good guesses as to what's wrong. Now, in periodic paralysis, the answer is almost always, well, there are several different types of issues, and in periodic paralysis, the answer is almost always one type of disorder, which is an ion channel that has one amino acid change to a different amino acid and therefore functions differently. But in the general question, there can be other types of things that can be wrong. One is called a deletion of a protein, and sometimes it occurs by a mutation causing the signal for stop making this protein to be put in at one particular point, and you tend to get only a tiny protein. Now, for a channel, as Dr. Lehmann explained before, that tends to be non-functional, so we don't. So those are typically horrendously affected, or only in situations. In the proteins that we're talking about in these various diseases, those you wouldn't see those. Another type, as he alluded to, are small or bigger changes in a chromosome that can affect several genes in a row, little pieces missing, and one would look for those with different tests. So often it's very important that a doctor know, do I order a specific gene test? Do I order something called a FISH test, which is not fishing, it's fluorescent in situ hybridization, but looking for a particular section of that you're suspecting. In some cases, there's a test called an array hybridization, where you basically are looking for lots of known genetic causes, and that's an automated thing on a chip. And so very often from the clinical situation, one can have pretty good guesses. Now the ultimate of this is basically going to be doing some combination of looking for these big changes and then looking at the whole sequence of the genome, or the express part, which is called the exome. And so what's the direction people are heading is coupling this up, so you compare the clinical part, which are the descriptions of each of the diseases, or what we call the phenotypes. So we call the whole collection of them the phenome, and comparing that to the whole genome. So it's a genome. You do a genome phenome analysis, which is basically within a decade or two. That's going to be a common way we're going to look at things. But we're not there yet. It's still expensive. We haven't put together the information of what these 80,000 differences mean, and how you decide which ones are important. But those are the ways we take clinical information and use it to be good guesses as to the genetics. And it's often very complicated because lots of things can go wrong, and when you have to prioritize 80,000 changes, you obviously have to have some rules of thumb.
But are there classes of drugs that you can, like once you know the gene, the exact gene, the mutation, then in your imagination of how the ideal scientific method works, will drug companies say, wow, cool, we've got a known mutation right there, let's develop something specific for this one?
So the question is, when you can categorize people by, you have this exact mutation, we know it to be pathogenic, can we decide on a treatment based on that? And the answer is, sometimes it will matter the exact details, sometimes it won't. But the more you know that information, the more helpful it is. And part of what goes on in studies is, and part of why certain studies will say we only want to include people with known mutations, is because they want to do exactly that sort of analysis. They want to say people with this mutation here, how have they done with this drug? Do they have these types of things? Because ultimately what we would like to do is be able to say, oh, well, based on the fact that you have this and that, we think you probably have this mutation in this channel, and then it becomes cheaper to look specifically for that. And then once you have that, the idea would be we have a pretty good idea of at least where to start on treatments. Now, it's not going to necessarily be everything. There are other things other than that mutation. It could be that it's going to depend not just on the specifics of your mutation, but it could be by whether you have some digestive problem, or whether you're male or female, or whether you tend to eat certain types of foods, or those sorts of things. So ultimately, we have to try to put all this stuff together. But yes, the more we know what your mutation is, the more we have specific information. That the hope is that even in situations where you won't be treated differently, at least we know what it is. And there are some situations where even 2 different genes produce the identical clinical picture. We're pretty close to that with periodic paralysis, in which we have a sodium and calcium gene that produce very similar patterns. But there are some areas where you basically get indistinguishable patterns from different genes. And we can obviously hand this back to Dr. Lehmann. I just was adding on to a previous question.
And if I may clarify, I guess my question is, so if you're having, and I think I'm understanding this way, if you're having genetic testing and you do not find a mutation in the known area, am I understanding that it is helpful then to have immediate family members, especially if you have somebody in the family also experiencing symptoms and ones that are not, also have their blood sent to be checked? Does that help in unknown mutations?
The circumstances there are 2 circumstances under which that's particularly helpful. One is if you found one of these 80,000 changes and it's not in your list of ones that cause disease, and it looks like it might, but you can't tell for sure, then, as Dr. Lehmann explained, then if all the people in the family who have it are affected and the ones who don't are not, then you think, okay, this really is pathogenic, and you add it to the list, the shared list that everybody has access to of what you think is pathogenic. So in those circumstances, it's really important. Having information from the whole family is very important in situations where you don't know the gene. So if you have a large family and you can pinpoint people, going up and down several generations who are affected, then you can do these statistical methods of looking at a whole bunch of things in a bunch of people, and then trying to see, okay, who has this part of this version of this chromosome and the ones, and you see what follows what. And for those circumstances, this whole penetrance issue, which people discuss, becomes very important. Because if you have people who don't show the symptoms, we're in danger of classifying them wrong. Yeah. And the more we classify people wrong, the more we make it difficult for Dr. Lehmann to get a good result, because he has somebody classified wrong, and that messes up all the statistics. So that's one of the reasons we're focusing on things like this lidocaine test, because the more you have various indications that can pick out somebody who has the gene but doesn't necessarily have the symptoms, the more that you can actually follow these things. And so in the example of attention deficit disorder, it's particularly important because 80% of people who are diagnosed as having attention deficit disorder are male. And the reason for that is that both males and females have the inattention part of attention deficit disorder, but almost all of the people who have the hyperactivity part are male, or at least far more than the females. So the long and the short of it is 80% of the males get 80% of the people diagnosed are males, and if you go through the math, that means that assuming that you're diagnosing correctly all of the males and not missing any of them, you're missing 75% of the females. So if you can then find something else, so it's hard to, inattention is a sort of hard thing diagnose with perfect accuracy because people can be different in various ways, and you can ask questions about what it's like to have a conversation with 10 people around you, and can you follow, but you're not going to get super accurate answers. So the more we can get things like when we put lidocaine on your tongue, does it do nothing or does it kind of numb it up? The more we have sort of really stark things like that, the more accurately we can follow the gene through the family, and then the more accurately they can crunch the data. Because for each error in terms of the way people are classified, it messes up his analysis even more. And it's very, there's the more you have the accuracy and the ability to say who actually has this, the smaller the samples, the faster the analysis, and all of that. So all of these things matter, and it's, we try to present it as being, as the more or less simple version, but there's all these complications. And so when, those of you who are in the 30% for hypokalemic periodic paralysis who don't have a gene known, you think like, what's taking these guys so long? This is what's taking us so long, and this is, so these are the kinds of things we're trying to do to just get a real biological handle on this. And the more you can do this, and typically in the history of these areas, when you actually get a really accurate biological handle on saying this person has it, this person doesn't, those are the innovations that lead to the progress in finding it, and then that leads to the progress in various other things. And it's particularly exciting with this, with the lidocaine-resistant people with hypokalemic periodic paralysis, because we think that they're going to be molecularly related to a common form of attention deficit disorder. So however common this variant is, and it's typically given as 10 per million for hypokalemic periodic paralysis, but I don't think we really know that number with great accuracy. For diseases that have dominant inheritance, we don't really have accurate numbers because we don't know how many people we're missing. For diseases that have recessive inheritance, which means that you have to have 2 copies of them, which are the channelopathies, I think are, all the ones I can think of at least, are dominant. Many other diseases are recessive, so something that's about as common as hypokalemic periodic paralysis, if it at least as common as people think it is, which is about 10 per million. So if you take something that has 25 per million incidence, the fraction of the population, and if that's autosomal recessive, which means you need 2 copies to have the gene, the fraction of the population who would have, who would be carriers is 1%. So it's pretty easy for recessive disorders. You check a few thousand people, and you know what fraction are carriers, and then you can calculate what fraction you'd expect to have the disease, assuming that people married each other at random, as opposed to within small ethnic groups and so forth. So we don't really know how common hypokalemic periodic paralysis is, but let's say it's somewhere between 10 per million and 100 per million. We think the channelopathy form of attention deficit disorder might be somewhere between 100 and 1,000 times as common. So if we can find this, it's going to focus a huge amount of interest on channelopathies and on hypokalemic periodic paralysis. And if you want something to publicize hypokalemic periodic paralysis, you're not going to find anything better than this. And this will become the disorder for, assuming that these hypotheses are correct, that this will turn this into something that every doctor knows about. And there's probably no other way I can think of to really get people's attention.
I have interstitial syndrome type 2. Dr. Linkenhorn, you have my blood, and Dr. Twill has my blood. How often is that good forever, that blood sample, or do you need to keep giving blood every so many years, or for further study down the road?
When we find the mutation, then we will never use this particular blood sample. That's only in cases where we're not able to find the mutation. In your case, it was found. So It's not ever used in the future.
So I don't ever have to donate any more blood to
Right,
you or anything
right.
else, we'll just keep using that.
Right.
Just pop up.
Yeah. If it's an accepted mutation, then it's clear forever.
What if it's not an accepted, the 30%?
If it's not accepted, then we try to get blood samples of as many as possible family members, and then to see is there a concordance between the clinical status and the genetic alteration we found. And if so, then the next step follows. We will functionally express this alteration and see if the channel shows a dysfunction which can explain the disease. And if so, then we think this can be accepted as a disease-causing mutation.
You can?
We'll do first before we do second.
Thank you. I've been wondering about, we've talked about the triggers to attacks, such as too much salt or high carbohydrate meal or something like that. But what about the triggers to the onset of the illness? Do we know much about that?
Yeah. So genetic alteration is lifelong. So from as a fetus, as an embryo, you have that, and all your life you have it. But it doesn't mean that you show clinical symptoms for all the time. The best example is periodic paralysis, at least in the state where only periodically or episodically come the clinical features. Normally, you are strong, you don't have a weakness, and episodically, after ingestion of carb-rich food, then you develop symptoms for a few hours. And that means usually the genetic or the effect of the genetic alteration will be coped or will be. Can you help me with my English? Yeah. Will be compensated for, yeah. And only if the compensation is not good enough, then you will see the symptoms and you will develop the symptoms. And that's also true for other diseases, let's say myotonia. Although the genetic defect is present, most Becker-myotonia patients show the first symptoms at the age of 14 or 15 years. So that means the first 14 years the genetic defect was compensated for, not on the DNA level, but on the functional level. That means either the gene wasn't expressed during the first 14 years and therefore no defect was visible in the cell, or other genes which are able to compensate this defect were expressed during the first 14 years. So there are many different possibilities to explain that the clinical symptoms occur early